2 in terms of size, difficulty, grade boundary. Then I'll give you a weekly plan on how to prepare in 15 weeks from now all the way till May. Then I'll show you how to prepare specifically for Unit 3. I'll show you where it takes to score an overall A in AS Biology. And finally, I'll show you the best study technique to achieve a lot, to get most of the things done before April. So by April, you could be able to start your revision. Now, here is a quick comparison between Unit 1 and Unit 2. First of all, in terms of weight, the number of row marks, and the length, Unit 1, Unit 2 are identical. There's no difference. The main blocks of Unit 1, which you're probably familiar with, are biological molecules and heart circulation. If you were to state the main blocks of Unit 2, they are mainly about cells, all sorts of cells, cell division, and it's about natural selection and evolution. In terms of the grade boundary to score an A, Unit 1 boundary is usually slightly lower. Every session you'll notice that Unit 1A is slightly lower than Unit 2, and that's mainly because students have already experienced the exam. Students have exam skills, so they enter Unit 2 and they're already taken at least the Unit 1 exam. So that's why they achieve better, and that's what drives the grade boundary slightly higher in Unit 2 than Unit 1. Now, in terms of topics, this is where I'm going to go into more depth. So I'm going to go with you week by week on how to prepare and what is the best order to go by. Because the syllabus, in my point of view, goes not in the best coherent passage through the topics. I mean, they start with cells, then they jump to cell division, then they come back to plants. So they keep going back and forth. The way I've done it over at least 10 years has proved to be way better. And again, end of the day, you will be getting done. You'll get the whole syllabus done in short time. You reach the same point, whether you go in by the same order of the specification or by this order. So first thing you start with, which will take you roughly 1 week, 1 lesson. It is the animal cells, plant cells, and what we call prokaryotic or bacterial cell. You do get a lot of questions here to compare between those cells, and they ask you specifically about the organelles. Like they ask you, what's the job of the nucleus? What's the job of the ribosome? And you're also required to draw, like literally you have to draw it with your pencil. You have to draw many of those organelles. In addition, this first week would also include this super important section. Not long, it's like half a page, but they do ask you this particular question a lot on how proteins are transported within the cell. We call this protein trafficking. How are they transported within the cell and how they're exported outside the cell. So this is like a continuation from where you started in Unit 1. So this is after translation. You did in Unit 1 transcription, translation. This follows that. Now you may ask me, do they ask me about things in Unit 1, in Unit 2? Small parts. I mean, you don't need to revise Unit 1 to enter the Unit 2 exam. You just need to solve past papers of Unit 2. And while you're solving them, you realize like there are small little parts of Unit 1 that come every now and then, but it's not worth revising the whole Unit 1 before the Unit 2 exam. So this was week 1. Now comes second week, which is here, and that's my third week, okay? So second week, it's gonna be all about plant tissues. So as you can see, I did the plant cell in the first week here. So this follows it perfectly, because now you're gonna learn about tissues of plants, including phloem, xylem, and another tissue I think you're not familiar with. It's just called sclerenchyma. So those are 3 plant tissues, the only ones you need to learn. You open the textbook, you'll find so many other tissues. No, only these 3 tissues are the ones that are required. Once again, what are the questions they ask you for? Especially xylem and sclerenchyma, they ask you to compare between them. Phloem, you just need to know its function and how it's done. So as I said, this is week 2. I still have one more thing, which is regarding the cell wall. And you can see now the sequence. Once again, we did cells here. Now it's time to do the cell wall. What do you need to know about the cell wall? You need to know how is it made, like the synthesis of the cell wall, which is another important question, and its structure, what is it made up, and its function. Now, if you know little of biology, you know that cell wall is cellulose, okay? Here, only in this part, I want you to revise the part about starch from Unit 1, okay? So here's cellulose Unit 2. They ask you to compare between those two, starch and cellulose. Starch from Unit 1, cellulose from Unit 2, okay? So that was week 2. It was all about plant tissues and about the cell wall. What about week 3? Week 3, still about plants, but this time it's about plant products. I mean, what are these important products that we make from plants? Mainly biofuel, and the second part are like fibers, clothing, ropes, and these things. And here's all about sustainability. I mean, like, how are these plant products way better than oil-based products? How are they way better than fossil fuel and plastics? Now, every time you see this sign here, that means that this is a practical. So this is my first practical of Unit 2. Probably know that the practicals could come in Unit 2 and Unit 3. So they may ask you about mineral deficiencies, magnesium and nitrate and calcium. Those 3 minerals are the only ones required. So they may ask you about this in Unit 2 and Unit 3. In addition, I have another practical, fibers, plant fibers. So we are gonna learn here their tensile strength. Means how can you test how strong those fibers are, and how can you compare these fibers to artificial man-made fibers like nylon. Okay, so that was week 3. All fall under the main block here, which is plants. So far, we started from last week of Jan here. That's where we started. Then first week of Feb, second week of Feb. So we're mid-Feb, and we've already done like nearly 30% of the content, okay? Now let's go to week 4. What's week 4 all about? It's about drug trials, drug tests. That's one of the most important, that's one of the most interesting topics, I should say. So what is this about? This is about how do pharmacists, scientists in general, develop new medicine. Think of vaccines, think of antibiotics, think of any medicine. What's the procedure that is done to perfect the manufacture of any medicine? How is this done? So we have several ways to make it, but here your main aim is to compare between we call historical drug trials. So how is it done in the past? I mean, long time ago. And how has it recently been done? So once again, they're going to ask you here to compare between those 2 methods. What are the advantages and what are the disadvantages of the historical drug trials? And how did we manage to perfect this method of drug development? Once again, we have our third practical, the most important so far. They do ask you a lot and a lot about this particular practical, which is the antimicrobial properties. This means like you need to test any plant extract, like you extract mint, garlic, onion, plant-based materials, and you test them on bacteria. It has a lot of details. It takes like nearly 30 minutes for me to teach it because it is quite very important. So that was week 4. You're done now with drug trials and you're nearly 50% done with this unit. Then comes a mini topic about classification. Did this in IGCSE. Here is like a bit more advanced. You classify all forms of life, not only animals, not only plants, all forms of life. You're going to classify these into 3 main domains. One we call eukaryotes, one we call prokaryotes, and the third one is the one you're going to learn in this unit. We call those archaea. So it's just like a modern way of classification. No longer you talk about like animal, plants, vertebrates. No, it's like an overview of classification of the entire life on this planet. Quite interesting, but not as interesting. That's my favorite topic. So that was week 5. Then comes week 6, 7. So what is this here about? This is gonna be about mitosis. You do study mitosis in depth, way more depth than IGCSE. So you're gonna study all the different stages of it. So just be careful. With mitosis, you need to learn all the stages in details. And you also need to be able to prepare a microscope slide where you study those stages. So that's my other practical here. Also quite very important. Not only for students taking AS, also comes for students who are taking Unit 6. That's like the last unit of A-level biology. Moreover, you need to do one calculation. We call this calculation the mitotic index. So that's math thing here. You need to know just one formula, very straightforward, but it's worth being mentioned. Okay, so that was week 6. Now comes week 7. As you can expect, mitosis, then meiosis. But with meiosis, you don't need to know it in details. Not like mitosis. No, here you just need the major events. There are just 2 main events that you need to focus on. And I'm gonna confuse you with the weird names. One is called crossing over. One is called independent assortment. When you reach there, you realize why they're called so. So then there is fertilization. You don't need to learn all about reproduction in humans or plants. No, just that part, fertilization. How is fertilization is done in plants and in humans. The events of fertilization, including structure of the sperm and structure of the egg cell. So this means we're 7 weeks ahead, and this means we are right here. So we did 1, 2, 3, 4, 5. Now it's 6, 7, right? We only have 3 weeks till we get to April, and we have decided that April is going to be the time where we start revision. So now, the last 2 things you need to know. One is called cell differentiation, also quite very related to Unit 1, mainly to gene expression. So here in this section, you will be following the events of translation, but in this part, you will learn how are genes turned on, off, based on the requirements. Briefly, it means that in your body, you don't have all of your genes active at the same time. No, you turn off those you don't need. That's called gene expression. And another favorite topic here is known as stem cells. So you learn all the ethics of using stem cells for therapy and for other uses. Okay, so this is week 8, right? Now comes the last part, or the last block, I should say, which is another week, week 9. Here is talking about biodiversity. So we are no longer talking about cells. We're no longer talking about organisms, individual. No, here we are talking about like environment, populations. So variation, like causes of variation, like you remember in IGCSE you had something called continuous, discontinuous variation. You do the same thing here. You also have the part about natural selection, and as you expect, there's evolution. In addition, you have a small section on how we conserve plants and animals. So this is the last block. Now, there is one part about microscopy. This is unit 2, but I personally teach this with unit 3 because it's all practical thing. What do you do with microscopy? So this is now, we're done with unit 2 completely. We managed to finish here by end of March. Now starts April, like nearly 6 weeks before your exam. By the way, if you don't remember or you don't know when the exam is, unit 2 exam is gonna be on the 20th here. That's unit 2 is the 15th, unit 3 is the 20th. So that's unit 2 and that's unit 3. Okay. So yeah, we've just started April and we're gonna kickstart April with microscopy. And as I said, this is mainly practical. And it talks about magnification. So one more calculation. You do conversions between different units like nanometer, micrometer, millimeter, etc. Calculations. And you need to draw. Now, if you've done Cambridge IGCSE, you had something called biological drawing. Even in Edexcel, there are drawings. So here you also needing to learn how to draw in the like biological way, a special way, drawing an outline, not sketching, not shading. Okay, so that's gonna be week 1 of April, but here you have to work smart because starting from April, you've finished unit 2, okay? And you've started unit 3. So while you're working on unit 3, here you will start to solve full past papers. So every week with my students, we'll be doing here combination of past papers. We do start from 2019 all the way to 2025. Every week we will solve 2 sets like 2019, 2020, 21, 22, so on. So every week we do 2 sets. So once we are reaching the last week of the exam, we're fully done with solving all past papers, every single one of them. And meanwhile, we're working on unit 3. Speaking of unit 3, what is this unit 3 about? So unit 3, it's no new content. So it's nothing you open like text or chapters about Unity. There's nothing new. It's just the practicals that you have done in Unit 1 and Unit 2. There are 10 of them, roughly. These are the practicals you need to work on. So starting from Unit 1, things like the beetroot experiment about cell membranes, the Benedict's, all about protein, food tests. These things are part of Unit 1 and Unit 3. You need to know basic theory questions, mainly about biological molecules. So they do ask you theory questions about starch, glucose, proteins. But again, I don't recommend you revise all Unit 1 before the Unit 3 exam because it's going to be a waste of time. While you're solving past papers of Unit 3, you will face these questions. They are repetitive, so you realize what are the things you need to remember. And the exam usually 3 to 4 questions. And there's always a graph, many calculations. And very important is that the Unit 3 exam is completely different before 2019 and after 2019. So I don't recommend you solve any old past paper because you'll find it totally different from the recent one in 2019 onwards. So what are these practicals you need for the Unit 3 exams? So you will need the ones about food tests like Benedict's. You need the one about enzyme rate of reaction, the beetroot, the DCP-IP. As I told you, microscopy is a chapter of Unit 2, but it's mainly needed for Unit 3. From here onwards, they're all practicals from Unit 2. The last one is not a core practical. It's not like the practical you'll find in bold in the specification. It's just some question about pollen grains. They do ask you a lot about in Unit 3 exam. So that's why I included here in the list. So you have 9 core practicals and that last one, which is just as important as the rest. Okay? So this means we're done with the first 2 parts here about the overview, the weekly plan, Unit 3. Now I'm going to start talking about the grade boundaries. How do you make up for marks you lost in Unit 1? How much you need to score in Unit 2, Unit 3, so that you can get an overall A, or if you're planning on taking A-level biology as a whole, what are the marks needed so that you can score an overall A star? So this is what the grade boundaries are usually are. Small changes every exam session, but they're not much different anymore. It's like nearly about these numbers. So you can see here the Unit 1 in this session is 49 out of 80 to score an A. It ranges between this much and like 56 out of 80 depending on difficulty. In this exam session, I believe the exam was quite long and the last couple of questions are not easy. So I'm expecting it to be within that range. Now, if you've lost marks in Unit 1 and you want to recover or you want to make up for those marks, how much you need to score so that you can take score an overall of A or A star in A-level biology. So just quick reminder about the weights of these units. You have 40% for Unit 1, 40% for Unit 2, and Unit 3 is half credit, it's 20%. This 20% is, you're gonna realize it is the hardest. Unit 3 is the one that requires a lot and a lot of solving. You really literally need to solve every single exam paper because questions are the most unpredictable among all units. It's usually Unit 3, Unit 6 are very unpredictable. So students who want to repeat a unit, I usually recommend repeating Unit 1, Unit 2 first because they have more weight, more percentage, and they're predictable, meaning you study, you get the score you studied for. But Unit 3 requires a lot of solving, and as I said, the questions come not as predictable as the other units. Now, for students who are taking A-level as a whole, then this time the unit 1 becomes half the weight, mainly because now you're going to be taking the other 3 units, 4, 5, and 6. So if you're just here, these are the weights for their AS. And if you're taking it as a whole for A-level, then it would be half. Which tells you something here, which tells you that if you're taking A-level as a whole, you can afford to some extent not scoring very, very high grades because you have more room to improve because you've taken 6 units instead of just taking 3. Now, unit 4, 5, and 6, they're just as much as units 1, 2, and 3 in terms of their percent. Okay? Now, speaking of UMS, because that's what matters to decide your grade, unit 1 is 120, unit 2 is 120, and unit 3 is going to be 60. Same applies to units 4, 5, and 6. Now, what it takes to score an A, as a UMS for AS first, you need 240 as a whole. So 240 out of 300 is quite not difficult to score. For B is going to be less, obviously, and for C is less. So A is, everyone, I think, study enough, solve at least half the number of past papers, you can reach to that 240. Obviously, you need to understand the content. But scoring an A star is where things become quite more challenging because you need that 480, which is 240 times 2, but there are 2 other small requirements. So you need that 480 I was telling you about, but for A2 specifically, you need to score 270 out of 300, okay? So again, if you're still in your AS, then aim for that 240. If you flopped your Unit 1 exam and you managed to score like a high B, mid B, you can still make it up with Unit 2 and 3. You score the C, it's not going to be very easy. It's going to be a bit more challenging. So B, high B, mostly it's very easy to recover that with Units 2 and 3. And if you're aiming for an A star, a low B, you must repeat the entire Unit 1 exam to reach to that, the 480. Okay? So this was briefly about the grade boundaries. Now, I just want to share with you how do you study, because till the moment people have done with the Unit 1 exam, still I see people, students, studying in the wrong way. And I'm not going to show you my way. Obviously, I don't study for this, but I want to show you how students who scored like full UMS, which are many I've seen, many I've taught, how did they manage to get the 120? These are how they reported their study method to me. So usually to get that full UMS, it's really not difficult. It's just you need to study the right way. So it's not about how much you study, how much time you spend studying. It's just like the way you funnel your time and effort. 4 things you need to do in their correct order, and I would guarantee you would score that full UMS. So step 1, your school teacher, your tutor, just take the lesson, and while learning the lesson, you just have to take quick notes. Usually with my students, I hand out like a PDF with the most important thing and just follow along so that you can grasp at least 40 to 50% of the details of the lesson. I'm not expecting you to get every single detail your teacher says. If you manage that 50%, you're good enough. You manage to reach that minimum level of understanding, which you will support right after the lesson. So I would recommend 2 hours, 3 hours, same day, maximum next day to summarize that lesson. That's one way, but again, it's time consuming, and most of the times I check their notes and they've missed the most important things. So either get like your teacher's notes or anyone's notes that you can rely on and go over the notes, underline, circle, write on the margins the things that you need to remember. So with the notes there, you have managed to boost that 50% you've learned during the lesson. You can manage to boost this all the way up to 70%, which would score you like a B or even a low A. But still, because we forget a lot these days, you need to support this now with step 3, where you're gonna solve questions, classified questions, only about this particular topic. But here is where everything changes. You have two types of students. Students who just want to get this done, and students who spend time going over the notes, checking their answers. Like personally, I check the answers of my students, but obviously you can always check your answers and mark the mistakes, mark the things that you got wrong. That's how you grow. That's how you improve. If you just solve, leave it, or don't go over your mistakes, or don't go over your teacher's like remarks on your homework, you're never gonna improve. You're just gonna be at the same level. You're never gonna exceed that 60%. So while you're solving here, you are actually memorizing and you're actually building those very valuable exam skills. Personally, I solve with my students the most important ones. I usually turn those questions into like fill in the blanks so that we could focus on the key terms. Because the best way to write answer in biology is to master those key terms and write a small sentence about each one. It's that simple. Okay? So how many steps we've done here? We've done 1, 2, 3. We could manage now 80% of the content. But again, you studied this topic in Feb, the exam in May, you have forgotten it. I guarantee you have forgotten everything about it. So as we've decided, starting from April, you're gonna start to link up the topics. So I take topics that are related, those blocks, and I know I've been doing this for more than 10 years. I know how they ask the questions. So I start to link up topics from all different areas of the syllabus. And here where things start to click. Here is where you reach that level of scoring an A by knowing exactly that cell differentiation is linked to Unit 1 and is linked to Unit 2. And somehow the question will come in this format. You can do this yourself. Just, once again, grab all past papers from 2019 to 2025. And again, solve effectively. Solve while highlighting the mistakes. You can only improve by marking your mistakes, highlighting them, filing them, and returning back to them like 1 week before the exam. That's how you avoid them during the exam. And that's how you get those like best perfect answers, you find them in the examiner report. You find answers are very well structured, nothing is missing, and the student's answers perfectly coherent. All right, so this was about the study technique. I think I have managed to finish on time. Now, if those of you were asking on whether they want to join like the normal classes, the live classes, there are live classes for Unit 2 and Unit 3 every Monday. From this Monday all the way to exam, we do one class per week first, and then April, the month of revision, we do 2 classes per week all the way till the exam night. So you have this option of live classes, but if you prefer, you can get all the classes in advance, including those revision, and you can join us again for the live classes before the exam. So both options are good. It depends really on what kind of person you are. If you want someone who like follows you up with the homework, with tests, take the first option. If you're responsible enough and if you could manage to study by yourself, then take that second option. Either way, you get all the notes, you get the solved past papers, and you get your work marked. Okay, I think that was it for today. Now tell me if you have any question. I'll be reading your questions here. If you don't have questions, then I'll see you next time.